In this video, we're going to be talking about how our body absorbs glucose, and then we're going to move into talking about insulin and diabetes. Not in a whole lot of detail, but just enough to give you a bit of an understanding. So, because glucose is the energy source for all of our cells, we are going to find glucose transporters throughout the body. That happens to be that we have different transporters in different parts of the bodies, and we're going to talk about that in the next couple of minutes. In most cases, glucose is going to be transported through the process of facilitated diffusion. If you remember from when we talked about facilitated diffusion, glucose is a polar molecule, so it's going to have some kind of help to get across the membrane, and in most cases it's going to go from an area of high concentration to an area of low concentration, which is what facilitated diffusion is. So there's actually at least 6 different transporters. There's the S-GLUT1, which is not a facilitated diffusion transporter. We already learned about it. It's the secondary active transporter of glucose and sodium. But then there's GLUT1 through GLUT5, GLU standing for glue, the T for transporter, and then they've numbered them 1 through 5. Fructose, which is very similar in structure, certainly in chemical formula to glucose, is going to be transported in through GLUT5 into certain structures. I should say different cells rather than different structures. And then galactose, again, another isomer of glucose, is able to get into the liver through GLUT1 and GLUT2, where it gets changed into glucose where it can be actually used. Galactose is not going to be used directly by our cells. So what I've done to help you remember them is I've included an image of the human body here with the various organs, and then pointed out where things are located. So GLUT1 is located, as you see, I have it pointed in a bunch of different locations. It's trying to give you the impression that it's in pretty much all of the cells, and that's what's going to be bringing in the amount of glucose that each cell is going to need to operate for whatever period of time. Also, you'll find it in red blood cells as well. GLUT2 is found in the liver and small intestines and in the pancreatic beta cells. We're going to talk more about the pancreatic beta cells because that's where insulin's produced, and glucose coming in through the GLUT2 transporter is going to tell the pancreas that it's time to release insulin. GLUT3 is a transporter up in the brain and neurons. And then we move to GLUT4, and GLUT4 transporters are actually transporters that don't appear so much on the surface of the cell until the cell receives the insulin's signal, and at that point in time the GLUT4 transporters are going to be brought to the cell surface to help bring glucose in out of the bloodstream to help your blood glucose get down to the level that it should be at. So it is found in muscle cells, skeletal and cardiac, and in the adipose tissue as well. And then GLUT5, as I said, was the fructose transporter in the liver. And I'm pointing out again the S-GLUT1, which we already learned about, which is that secondary active transporter that helps to bring glucose in from the glucose that we've ingested into the intestinal tract, or I should say into the intestinal epithelial, so that it can then be spread throughout the body where it's needed. So insulin is a protein-based hormone. We have other hormones that are not protein-based, more of steroid-based. I've shown you here the structure of insulin. I don't expect you to memorize it, just trying to give you an appreciation for it. There's an alpha chain, there's a beta chain. You see these lines with S's on them, those are disulfide bonds. That helps to give the insulin the structure that it needs. And as I was pointing out with the GLUT4 transporters, the insulin's going to signal to those cells that it's time for them to bring in more glucose than they normally do. As you probably already know, insulin is what's produced in response to the signal, which is high glucose in the blood. So whenever you eat a meal, your insulin is going to be released to help bring down glucose levels. Of course, the more glucose you eat, the more insulin is going to be produced to try to bring that level back down to an appropriate concentration of glucose in your bloodstream. But insulin, besides being protein-based, is going to need some processing in some of the cell organelles that you've been learning about on your own, and we'll go through that later on. And then it's going to be secreted from the pancreatic beta cells to get into the bloodstream and then head to the target cells to tell it it's time to bring those GLUT4 transporters to the surface. So here's an image of part of the cell structures that you were learning. You probably recognize this as being the nucleus. And we're going to start there. So when the pancreatic beta cells, this is what we're talking about, when they get the signal that the glucose is too high, they are going to cause any insulin that's already been produced to be secreted, to start bringing down the glucose levels. Also, at the same time, while that's happening, the nucleus has gotten, because it's measured the amount of glucose that at least the cell has, the nucleus gets the signal to start transcribing the insulin gene. And if you don't remember what that is, that's where the RNA polymerase reads the DNA to make the pre-messenger RNA for the insulin gene. So then we get that after the signal's generated, the transcription takes place. That's our second step. The third step, still happening in the nucleus, is we have the processing. We talked about messenger RNA splicing, where the spliceosome removes the introns and puts on a cap and tail. That's done here in the nucleus as well. And as a result, we get a mature messenger RNA that's ready to leave the nucleus, and it's going to come out through the nuclear pore that I'm showing you right there. So after the messenger RNA has left the nuclear pore, it goes to one of these ribosomes that's embedded on the surface of the rough endoplasmic reticulum, and it gets translated. And as it's getting translated, the protein actually gets injected, so to speak, into these spaces here within the rough ER, while the messenger RNA just slides through the ribosome on the outer surface of it. So that's the fifth step, the translation. While it's in the rough ER, there are going to be some enzymes that are going to put sugar groups onto insulin, and we use the term glycosylated. And what's interesting is in the biotech field, where they cloned the insulin gene and are now producing human insulin for diabetics, they are producing it in most likely yeast, which is a eukaryotic organism like we are, because it will glycosylate it correctly. If you try to produce this particular protein in bacteria, bacteria do not glycosylate, or at least they don't glycosylate in the same manner in which eukaryotic cells do. So it's not going to be effective. In fact, it actually could cause an immune response, possibly. So we're going to be using, or in the biotech industry, they're going to be using a eukaryotic cell to produce the insulin. It used to be in old time, before they had genetic engineering, that sort of thing, they would have to isolate it from, usually they isolated it from pigs, because they were closest in structure to our insulin. So then the seventh step, still in the rough ER, is that insulin gets folded into its proper structure. And I was showing you that on the previous slide, where there was some disulfide bonds. And then at that point, the insulin gets packaged into a little membrane ball that we call a transport vesicle, and it's shipped off to the Golgi apparatus. So I repeated the step number 8. Here's that vesicle containing the insulin. It's going to get to the Golgi apparatus, and it fuses with it. Now the insulin is inside the spaces within the Golgi apparatus, and then shipping information gets attached to the insulin. This lets the Golgi apparatus know where to put the insulin and when to ship it out of the cell. So that's an essential function. And then finally, when it's time to secrete it, the insulin gets put into a secretory vesicle, and that secretory vesicle is transported to the plasma membrane where it will fuse and then get out into the fluid around the tissue and ultimately into the bloodstream. And that where it's fusing with the membrane, that we call that process exocytosis, which we talked about in a previous lecture.